Well, thank you for joining us today for Dr. Ahmad's presentation on redox status in workers occupationally exposed to radiation. Dr. Ahmad is a faculty member in the MITS division, and so we thank her for joining us today. The majority of her scientific career has been studying the role of oxidative stress in the context of cancer biology, infection, and radiation. She has a broad background in redox biology with specific training and expertise measuring the enzymatic activity of various antioxidant enzymes. The research study she's going to talk to us about today focused on understanding the redox status of persons who are occupationally exposed to low-dose radiation. The study was funded by the Department of Clinical, Diagnostic, and Therapeutic Sciences through a Charles O'Malley Foundation grant. So with that, I'll let Dr. Ahmad begin her presentation.
Thank you, Laura. Hello, everyone, and thank you for coming today and listening to my talk. So today I will walk you through the research that I have conducted since 2014. Let me use first my highlighter. That has been conducted since 2014, and I will talk today about the redox status in workers occupationally exposed to radiation. So these are the topics that I will talk about today. I will give you a brief background about the biological effect of ionizing radiation, a little review about different sources of reactive oxygen species, and we'll talk about what do we mean by oxidative stress and antioxidant, and then give a little literature review about the topic. And then I will move and talk about the research design and methods, discussing the goals and showing you the results of this research, and then ending with conclusion and future directions. So the main focus of this research is looking at the long-term effect of exposure to radiation. Although X-ray workers are not exposed to X-ray beam directly, they receive scatter radiation reflected from their patients as well from the wall where they stand behind to an extent that is extremely variable. These doses of radiation exposure have some biological effect whose intensity and relevance are, however, complex to predict. And so the biological effects of ionizing radiation, they originate primarily from the effect to the critical target molecule, which is the DNA. And the type of the effect that radiation can have on the DNA is determined whether you are exposed to a high dose radiation or low dose radiation. So high dose radiation exposure, it will mainly kill the cells. And so you will have an immediate and measurable effect, and these are known as a deterministic effect. They have a threshold to occur above 150 millisievert, and their interaction with the DNA is directly as shown on this sketch diagram, where it hit the DNA directly and leading to that damaging effect. These effects are manageable as recovery and repair can occur. However, with a low dose radiation, here the effect is not, we don't see any killing effect. The only what you see is mainly damaging effect to the cell, and it depends on which cell they are damaged in order to see the effect. For example, if the somatic cells are damaged, you will see cancer. If the germline cells are damaged, you will see a hereditary or genetic effect. Exposure to these doses of radiation, they will have no immediate noticeable effect, and it will take so long in order to be shown. And that's what worried us as a researcher. And these are considered as a random and stochastic effect, where the probability of the effect is dose related. These stochastic effects are worrisome as they include cancer and genetic effect. Their interaction with the DNA is indirectly through the radiolysis of the water that lead to generation of free radicals that can propagate and hit and cause damaging effect to the DNA. So the doses to which X-ray workers are exposed to actually is not high enough in order to cause these deterministic effects, which as an example, an erythema and skin damage that have been noticed in radiation therapy patients who are receiving a very high dose of radiation. And as a result, these workers, they are continuously and considered their dose of exposure is a low dose radiation, which is the main focus of this research. So then what makes the link between radiation exposure and cancer? There was an increased incidence of certain types of tumors that has been reported and linked to radiation, and those were observed from human experiences, whether early or later, and these are an examples. So as an example from early human experiences, skin cancer has been noticed in early X-ray workers, and that's due to the lack of radiation protection at that time. Lung cancer also have been observed in uranium miners. The same thing with bone cancer observed in radium dial painters. Now, from later human experiences, we have seen that leukemia have been has an elevated levels in survivors of Hiroshima and Nagasaki. Same thing with patients who were treated for their ankylosing spondylitis. They received radiation and they end up dying from leukemia. And elevated level of leukemia also has been noticed in early radiologists since 1922, and that's due to the lack of radiation protection at that time. Thyroid cancer also have been observed in children who were treated for their enlarged thymus, and the same thing with the women who were treated with radiation for their tuberculosis and end up having breast cancer. Our organization, ASRT, American Society of Radiologic Technologists, has teamed with University of Minnesota and as well with National Cancer Institute, and they followed more than 90,000 current and former radiologic technologists. And they're looking at, they were looking at the long-term effect of radiation exposure. And this is one of their studies that they have been published, and where they see a twofold increase in the brain cancer mortality in the radiation workers who worked with interventional fluoroscopy versus none. And as you can see, there was an a modest increase in the incidence of breast cancer as well as melanoma. However, there was no increase in other type of cancer such as rectum, lung, colon, and so on. The only missing information in this paper that the researchers in this paper, they did not do a dose-response relationship. They did not measure how much dose these workers received in order to make that definite link between the radiation exposure as well these effects that have been shown here. And so the mechanism mediating these phenomena for radiation-induced cancer are complex and likely involve elevated levels of reactive oxygen species, DNA damage, and immunosuppression triggered by ionizing radiation. Although the mechanism mediating these phenomena is complex and incompletely understood, it is believed that production of reactive oxygen species triggered by ionizing radiation appear to play a central role. And it is well known that radiation can induce reactive oxygen species such as hydroxyl radical, superoxide, hydrogen peroxide, and other hydroperoxide. Several studies have shown a potential for these reactive oxygen species in inducing mutation, DNA damage, as a predisposing factor for cancer, diabetes, and age-related disorders. Now, what do we mean? Then we ask ourselves question, what are these reactive oxygen species? How can we protect ourselves? And what is the significance actually for studying this? So reactive oxygen species and free radicals are considered a toxic byproducts of cellular metabolism. So during respiration, we have an oxygen around, we use oxygen, and the oxygen will be around the mitochondria. And so normally, there was a 5% leak of electron from the mitochondria, which can be captured by a molecule oxygen, and this can lead to generation of the superoxide free radical. So as you can see in this sketch diagram, the superoxide is a precursor free radical that can lead to subsequent generation of other damaging reactive oxygen species, such as hydrogen peroxide, as well hydroxyl radical. I want you to pay attention that all these are considered reactive oxygen species, which means that they are species of oxygen that are very reactive than molecular oxygen. However, superoxide and hydroxyl radical are the only one that are considered free radical, as they have one unpaired electron in their outer shell. So what are then the sources of these reactive oxygen species? Actually, we have endogenous and exogenous sources. The endogenous sources of RRS are NADPH oxidase, which is a membrane-bound enzyme that is passing the electrons from the NADPH and NADH to a molecular oxygen that can lead to generation of superoxide and then to subsequent generation of other damaging species of RRS. Xanthine oxidase is found abundant in the liver as well in the cytoplasm, and it can, as it passes the electron from xanthine and hypoxanthine to molecular oxygen, this leads to generation of superoxide as well the other damaging species here. As we mentioned before, mitochondria normally have 5% leak of electron that can be captured by molecular oxygen, and it can lead to generation of superoxide as well as these bad damaging species that can lead to an oxidative damage to the cellular biomolecules such as DNA, lipid, and protein, as evidenced by their oxidative damage product, which is 8-hydroxyguanosine for DNA damage, lipid peroxidation, which is 4-hydroxynonenal, and malondialdehyde for lipid peroxidation and damage, and the protein disulfide as a damaging product for the protein oxidative damage, okay? Now for the exogenous or environmental sources for these damaging species, it can come from radiation, from pollution, from stress, as well from poor nutrition. All of these can lead to generation of these bad guys, which are the reactive oxygen species as well free radical, that can lead to consequences such as cancer, aging, heart attack, diabetes, dermatitis, and other health complications. The good news that our body is equipped with antioxidant defense system. And for the purpose of this research, we are focusing on the major antioxidants that are in our body, such as superoxide dismutase, SOD, catalase, as well as the glutathione system. So the superoxide dismutase, it converts the superoxide into other damaging species, which is hydrogen peroxide. And then hydrogen peroxide is converted into a harmless water molecule by other antioxidants such as catalase and the glutathione system. That doesn't mean these are the only antioxidants in our body. We do have other antioxidants such as vitamin E and C, but our focus is on these three major antioxidants. In our body, at normal situation, there is a balance between the oxidant and the antioxidant. When this balance is disturbed, and then what will happen is an oxidative stress damage will occur that can lead to deleterious effects. So our workers, the workers working with radiation, they are exposed to a long-term low doses of ionizing radiation. And although the effect is not seen immediately, it will, the probability of this effect to be shown, it will increase with increasing radiation doses, and that's what worried us. Previous studies confirmed the existence of oxidative stress in these radiation workers, as well. They showed that it might lead to DNA damage and mutagenicity. And there was also a growing body of evidence indicating the immunological changes induced by exposure to ionizing radiation. However, the specific ROS driving the oxidative stress in X-ray workers is not yet studied, and we are the first one who studied that. The other thing, there has been a little research examining the dose-response relationship of ionizing radiation, induced oxidative stress, DNA damage, and immune alteration, and also how these effects can change with different occupational settings were not studied before, and that's why it remains to be elucidated and need to be investigated more. Therefore, we want to take it one step back, looking for biomarkers that are thought to play a central role in development of cancer. And so oxidative stress, DNA damage, and immune response level are can be used as a predisposing factor for cancer, and those biomarkers could be of importance to guide health promotion and disease prevention, and more specifically in occupational setting with a high volume of radiological activities, as well with a high doses of radiation. Now, by understanding that superoxide is a precursor free radical leading to the generation of other damaging ROS species, so the goal of our study has been divided into 3 phases. So in phase 1, we want to assess the alterations in the redox environment at different occupational settings. And we choose in this phase workers who work with diagnostic radiography at different occupational settings, such as conventional radiography, computed tomography, and interventional radiography. And then in phase 2, we use the same cohort where we assess the downstream effects of long-term exposure to ionizing radiation. And in phase 3, we decided to assess the redox status in workers occupationally exposed to ionizing versus non-ionizing radiation. Of course, all our results compared with an age and sex match control individuals. So for the method that we use, in phase 1 and 2, we recruited about 20 healthy exposed workers working in diagnostic radiography, where are at different occupational settings such as CR, IR, and CT. And we also have a 40 age and gender match controls. In phase 3, we increase our diagnostic radiography accrual from 20 to 43. And then we recruited from nuclear medicine 7, and we have 8 volunteers from radiation therapy as well from ultrasound. And also we increased our control subject from 40 to 60. All our subjects are from Nebraska Med as well from UNMC, and our study is IRB approved. All volunteers have completed detailed questionnaire concerning their habits, lifestyles, and medical records such as age, medication, smoking, and so on. We collected their blood samples, and then we measured these biomarkers at different level. So in the whole blood, we measured superoxide using electron paramagnetic resonance. In red blood cells, we measured the antioxidants, SOD, catalase, and glutathione. In the plasma, we measured lipid peroxidation, DNA oxidation, cytokines, as well the extracellular level of SOD and glutathione. Let's now move and talk about the results. And in phase 1, where we looked at the redox status in workers occupationally exposed to long-term levels of ionizing radiation. So this table is showing the demographic characteristics of the study subjects in both groups. And as you can see here from this results, we have a comparable baseline characteristics in terms of the age, gender, alcohol consumption, smoking habit, dietary supplement, as well as their exercise level. Also, in this table, I'm showing you the mean dose of radiation that these workers annually receive. And as you notice here, it's about 2 millisievert, which is within the limits recommended by the International Commission of Radiological Protection, which is less than 5 millisievert per year. So the first thing we looked at is the superoxide level. And as you can see in this graph, we have a significant increase in superoxide level in radiation-exposed worker as compared to the control, with a marked differences among different occupational settings, as shown here in this sample spectrum for ABR, as well in this data summary. So as you can see, this marked differences has been more noticeable. Excuse me. It is more noticeable in IR and CT subgroup as compared to the control and CR. However, we did not see any significant changes between the CR and control, and the same thing between the IR and the CT. And then we measured the plasma lipid peroxidation. And as you can see, we see a significant increase in the lipid peroxidation in radiation-exposed workers compared to the control, with more noticeable effect in IR and CT as compared to the control and CR, where we did not see any changes between control and CR, as well, no differences between IR and CT. So with this increase in superoxide level, as well as in lipid peroxidation, that suggests that there is an oxidative damage on the blood of these workers. And so we thought about measuring what will happen then with these antioxidant level. So we measured the intracellular level of SOD. And as you can see, we see a significant increase in the intracellular level of SOD in radiation-exposed workers as compared to the control, with a marked difference among IR and CT as compared to the control and CR, where there's no significant differences between control and CR, and there is a significant difference between IR and CT here. So this result actually is very, very interesting. So with the increase in superoxide level and increase in lipid peroxidation, that indicates an oxidative damage on the blood of these workers. However, with that increase in intracellular level of SOD, this suggests that there is an intracellular protective mechanism, which is really a good sign, okay? Now, as I mentioned, with considering that increase in superoxide as well increase in SOD level, then it is tempting to speculate that the level of hydrogen peroxide will be elevated as well, as hydrogen peroxide is a product of SOD catalase dismutation of superoxide. Although we did not measure the hydrogen peroxide level directly, we did measure the antioxidant level that are involved in the detoxification of these hydrogen peroxide. So we measured the catalase level as well as the glutathione. And as you can see, there is no significant differences in either one between both groups here. And then we looked at the lifetime effective radiation doses that these workers have at different occupational settings. So although we did not see a significant differences between the dose level that these workers are receiving, however, you can see a trend of increase from conventional radiography to interventional radiography to computed tomography. And then we decided to create a dose-response relationship for the superoxide level as well as the lipid peroxidation. As you can see in both, we can see that there is a threshold dose-response relationship where we have a threshold at 17 millisievert, which is equivalent to the CR subgroup. And this response increase as we go beyond that threshold dose, with lack then of linearity had been shown as we go beyond of 31 millisievert, which is equivalent to the level of the dose received by the IR subgroup. And this is shown in both curves. This result is incompatible with the linear no threshold model which propose a proportionality between the radiation exposure and cancer. And so we have to interpret this result cautiously. Also, we saw a positive correlation between the superoxide level and lipid peroxidation as shown here, as well with the SOD activity. So in this phase 1, what we can conclude that radiation exposure within limits recommended by the ICRP can result in an altered redox balance, which is evidenced by an increase in the superoxide level as well as increase in lipid peroxidation. and also accompanied by an enhanced level of SOD as an adaptive cellular response. And then we decided to move on and continue our work for phase 2, looking at the downstream effects of long-term exposure to ionizing radiation. So we measured the cytokine levels, and we screened about 30 cytokines as shown in this table in the plasma level of both control and radiation exposed workers. And you can see we only see significant differences in radiation exposed workers as compared to the control only in IL-6 as well as in MIB-1 alpha and as well in IL-1 alpha. This table here, it shows you in which occupational setting these differences to these cytokines are found. And as you can see here, IL-6 and MIB-1 alpha showed a significant increase in their level in IR subgroup here as compared to conventional radiography as well as to the control. And although the CT subgroup did not show any significant changes as compared to both conventional radiography and control, however, there is a trend of increase if you compare and look carefully to this table, okay? So also, we did not see any changes between these cytokine levels between the control and the CR, as well as between the IR and the CT. Of these 3 cytokines, only the MIB-1 alpha showed a positive correlation with the superoxide level, okay? Now, as I mentioned, and as I mentioned before, and what we have shown with the superoxide level, when we did the dose-response relationship, we saw that threshold dose-response relationship. We did the same thing with MIB-1 alpha. We created the dose-response relationship for MIB-1 alpha, and we saw the same trend, which is a dose-response relationship, which is incompatible, again, with the LNT model, and that has to be interpreted in cautious. Then we looked at the DNA oxidation. As you can see here, we did not see any significant differences between the radiation exposed workers and the control. And the same thing, we did not see any differences between different occupational settings. And then we measured the extracellular level of these antioxidant, and we looked at the SOD as well as the glutathione level. And as you can see here, we did not see any changes in either SOD as well as in the glutathione. The results for the extracellular SOD and seeing no differences between the radiation exposed worker and the control is really very interesting. If you remember in phase 1, we showed you an increase in the superoxide as well as accompanied by an increase in the intracellular level of SOD. And however, we did not see any differences in the extracellular level of SOD. So these results suggest an intracellular protective mechanism to counteract the increased level of superoxide level and not extracellularly. So then we can conclude from this part that there is a dose-dependent relationship with significant immune alterations in radiation-exposed workers, and more specifically in IR subgroup, which are exposed to higher doses of radiation. And although the biological significance of changes in these cytokines is unclear, results from our current study indicated the importance to take all necessary measures in order to protect radiation workers from radiation. Now, the underlying mechanism of the cytokines elevation, it still needs to be further investigated. And then we move to the phase 3, where we recruited other occupational workers from other occupational settings, such as nuclear medicine, radiation therapy, as well ultrasound. And we want to assess the redox status in workers occupationally exposed to ionizing radiation versus non-ionizing radiation. And here is the results. So as you can see here, we measured the superoxide level, and by looking carefully here, how the superoxide level changes at different occupational setting, we see a significant increase in diagnostic radiography as compared to the control, as well as in ultrasound. However, we did not see with nuclear medicine and radiation therapy, although there is a trend of increase in nuclear medicine here. When we look to the SOD level, which is the intracellular level of SOD, we see an increase in the level of SOD in all different occupational settings, with a more significant differences in nuclear medicine and radiation therapy as compared to the control, as well with the ultrasound. And then when we tried to look at these results in different way by pooling all occupational settings that use ionizing radiation together and compare it to the non-ionizing radiation, which is ultrasound, and see how the effects are different. And this is what we get. So when you look to the superoxide level, you can see that the superoxide increase in both IR and the ionizing radiation and non-ionizing radiation group, with more significant differences in non-ionizing radiation as compared to both ionizing and the control. And the same thing with the intracellular level of SOD. We see a significant increase in the level of SOD in ionizing radiation group, as well as a non-ionizing radiation group as compared to the control, with more significant differences in the non-ionizing radiation as compared to both ionizing as well as the control. Also, we continued and measured other antioxidants that we believe that it can change and see how they are differ. So we measured the catalase activity as well as the glutathione level by measuring the ratio of reduced to oxidized glutathione. And when we looked at the catalase activity here, we see in all different occupational settings, you can see a significant increase in the level of the catalase as compared to the control. And when we pull these ionizing radiation together, you see that ionizing radiation group have a significant increase compared to the control. Same thing with non-ionizing radiation. However, non-ionizing radiation is more significant as compared to the ionizing radiation group. When we look to the ratio of reduced to oxidized glutathione, also what we see, there's no differences between diagnostic radiography and control. However, with other occupational setting, you can see a reduction in this ratio, which is an indication of having an oxidative stress. And when we pull these ionizing radiation group together, you can see that with ionizing radiation and non-ionizing radiation, there's a significant decrease in the level of the this ratio, and to more extent level in the non-ionizing radiation as compared to the ionizing radiation group. So based on these results that we have shown, and this is done during the pandemic period actually, and we want to continue and work more and looking to the white blood cell level of antioxidant and other things. But this time we can conclude from this part that chronic exposure to ionizing radiation as well as non-ionizing radiation has been shown to be associated with an altered redox balance, as evidenced by an increase in superoxide level, as well as a decrease in the ratio of GSH over GSSG, and that's an indication of having an oxidative stress damage in the blood of these workers. However, we saw an adaptive cellular responses, which is evidenced by an enhanced level in the antioxidant defense system, such as increase in the SOD level, as well as increase in the catalase level, in order to counteract the increased level of these ROS stress. Now, these effects have been shown to be more noticeable in the non-ionizing radiation as compared to the ionizing radiation. So, of course, our study has some limitations. The first one, a larger sample size would allow us to have more statistical significance in evaluation of these biomarkers, and that's our future to increase our sample size. The other limitation we have is that using that 8-hydroxyguanosine to estimate the DNA damage is another limitation, as this plasma hydroxyguanosine level can be influenced by the rate of repair as well as by the rate of damage. And so the information about the exact white blood cells profile in our samples was not provided, and in order to make a strong conclusion regarding the precise source of superoxide. These are the limitations in our study. Now, our future direction is that we want to take a closer look for mechanistic increase in these three cytokines, IL-6, IL-1 alpha, and MIB-1 alpha. And also, we want to look at a redox parameter across a much larger population of radiation workers who work in ionizing radiation and non-ionizing radiation by recruiting more. We need more number in non-ionizing radiation by recruiting more from ultrasound as well as from magnetic resonance imaging. And also to assess the level of the redox parameter during 2 periods of time, during the normal working condition, as well after taking some time of vacation, because we believe that with vacation, you can mitigate these effects, and that's what we wanna look at and see what recommendation we can give these workers. So at the end, so I'm done. This is what the study that I have done so far, and looking more. I hope we have more to come, and I would like at the end to acknowledge all the people who are involved in this research. Big thank to Lisa, who's my program director and who helped me to get the support from the O'Malley Foundation. And of course, Dr. Zimmerman, who provided me the lab space to do my research, and Dr. Adam and Dr. Maher Abdullah, as well as I would like to thank workers who volunteered to this research from Nebraska Med and UNMC, and big thanks to James Railing at Radiation Department, who helped us to spread the message of this kind of research and have volunteers in this study. As well, I would like to thank the Clinical Research Center, who helped us to collect the blood samples and separate the plasma from the red blood cells. With this, I'm done and looking forward to any questions if you have. I don't know how good I am done with the time.
Dr. Ahmad, this is Wayne Matthews. Very fascinating work on the effect of specific cytokines. And knowing that CD8 cytotoxic T cells secrete most of those cytokines, have you seen any correlational study on how ionizing radiation would affect the T cell balance, CD4 to CD8, and the CD8 functioning?
Thank you. This is really a good question. Actually, I did not look at that, and that's the hope that we are looking for in the future to look at, and that's a good question. Thank you so much. Actually, I'm hoping, but that I think I'm not the expert in the cytokine, so if you are willing to contribute, that would be good. But we are looking forward to look at that. Thank you.
Great, thank you.
Any other question? Laura, you are muted.
Thank you. Second time today. Excellent presentation. That was fascinating. I have a question about when you did the ionizing radiation and the non-ionizing radiation. What was your N? How many subjects did you have in the non-ionizing radiation group?
Yeah, it was actually shown in the slide. So in the non-ionizing, which is the ultrasound department, so we recorded only 8 subjects, and that's the number we get. And with this small number, we saw that big differences as compared to the other occupational. But we are hoping. Actually, we started that during that pandemic time, and we hope in the future to have more volunteer from ultrasound. And I'm hoping to collect as well from MRI, the magnetic resonance imaging, and ionizing radiation.
Did you evaluate the data to compare other demographic factors that could affect some of those some of those oxidative stress measures like BMI or their activity level, just to see if there was any significant differences between groups since you had small numbers. Was there any stark differences between the groups in that?
Actually, yeah, actually, we collected the demographic characteristics of these study subjects, and we did not see any differences between both. They are really matched. So we didn't see any significant differences between these characteristics. And so with that said, then, and using matched group, then whatever changes in the results that we have seen will be due to radiation and not to these confounding factors. But I did measure the BMI, but we can, and I asked people and they said, I can measure that and to check specifically for the BMI level. But if you remember in that demographic characteristic table, if you want me to, we did, and we found a comparable baseline characteristics. But this is
I didn't know if that included the ultrasound workers.
No, this is not. No, this is not. Yes, this is only for phase 1 and phase 2. And to be honest, I did it for the other group where in phase 3 we have nuclear medicine, ultrasound and radiation therapy and diagnostic radiography. I didn't see any changes, but because of the time, I didn't finish, like I did the stats, but I didn't create that table to show it here. We did not see differences. Yes. Thank you for that question.
I have another question, but I don't want to take time from everybody else. Okay, I'll ask my question. Did you hypothesize that the non-ionizing radiation workers would be significantly different than or higher than the workers in ionizing radiation?
You mean the level of these biomarkers?
Right.
Yes,
Were those results?
Yes, we did.
You expected it that way. Okay.
Oh, you mean, no, I didn't expect it that way, to be honest. I was expecting, because with ionizing radiation and non-ionizing radiation, both of them interact with your body different way. So ionizing radiation, they create ion pair through radiolysis of the water, and that can lead to generation of free radical. So I'm expecting to see more like oxidative stress, more oxidants and changes in the redox balance. However, non-ionizing radiation, they interact with your body, as the name implies. It's non-ionizing. They only do excitation. And this excitation, it is like thermal energy it will create. But when and then when I saw this result, I was shocked, to be honest. But then I did my search, and I found many papers mentioning that the non-ionizing radiation, they can lead to generation of the reactive oxygen species. So that is really interesting. And so we hope we can get a paper out of that. So, but I need to verify that by increasing the number of the volunteers, and hopefully it can be another paper. Thank you.
Yeah, fascinating.
Thank you.
Hello. Very interesting talk. I am from biochemistry department. I'm a glycobiologist. Recently, I became interested in the potential, the effect of stress. And one of the endpoint I am looking at is about infection. And I'm just wondering if you have come across any information about that. Infection like pneumonia or the GI problem with the people you are studying?
That's beyond actually my expertise. So as you heard Laura talking, my expertise is in redox biology, looking at the antioxidant and oxidative stress. But I know that our organization, the ASRT, they looked at, for example, having hypertension, heart attack, maybe other diseases that is related to radiation exposure, but I have not looked at that.
I'm just wondering if there's any record of your volunteer, study subject have those information.
I didn't look actually. No, I didn't include that in the questionnaire. Maybe that's a good point. Yeah, because we want to see something that, because I mean, when you look at these workers, you say, okay, they're normal, but
Right.
the concern that I have right now, I wasn't focused on inflammation or infection. My focus was on radiation-induced cancer. And many studies prove that long-term and repetitive exposure to low dose of radiation can increase your risk for having cancer. And that's why I want to take it one step back, looking at these biomarkers that I told you, to see if there's any changes and if we can have any protective measure or any, anything we can do. But that's the point, like looking at pneumonia. You are right.
Sure, I understand that. But pneumonia or this GI issue, much easier to monitor than the cancer. So as an endpoint.
Okay. Oh, thank you.
Thank you.
Thank you. That's a good suggestion. Thank you.
Any other questions? Well, if not, thank you.
I can see Laura, there's chat, but I don't know. Oh, okay. I thought
They're all congratulating you for an excellent presentation. So
okay, thank you. Yeah, thank you.
I guess with that, we will, you should read those before you log off. So before I close the session down, I'll make sure you read the chat, Ahmad, because we have multiple congratulations and praise for our presentation today. Thank you.
Thank you so much.